there is this misunderstanding by some, including Elizabeth Blackburn, that telomerase and long telomeres cause cancer. And what I want to explain is what they're misunderstanding. Why is it they're misunderstanding this? Because it's not the first time that they have actually misunderstood this. Originally, it got started because remember I told you that my team showed that if we inhibited telomerase with the antisense, we inhibited telomerase, we killed cancer cells. Well, a lot of people said, wow, if inhibiting telomerase kills cancer, then telomerase must be the cause of that cancer. But that was discredited years ago, simply on the basis that's like saying glucose causes cancer because cancer grows better in the presence of glucose. It wasn't. And as Dr. Raphael did a very good job of explaining the difference between something helping a cell to be healthy versus actually making it into a cancer. But then people, including Elizabeth Blackburn, started saying, well, why did we evolve telomere shortening? When we know now that our reproductive cells produce telomerase. It doesn't make any sense that we would then shut that gene off to produce telomerase so that when we grow up, our telomeres get shorter. Why would we do that? There has to be some evolutionary benefit to doing that. And what they said, it must be an anti-cancer mechanism. This is where Dr. Raphael said I would probably challenge him. I believe from an evolutionary perspective that telomeres are long enough. This is not just a belief, this is a fact. Telomeres are long enough that if a cell in your body gets cancer, that cancer will kill you many times over before the telomeres get short enough to actually senesce. So even in the absence of telomerase, the cancers are still going to kill you. And that's one of the important things to remember. It's not an anti-cancer mechanism. In fact, from an evolutionary perspective, evolution doesn't want us living longer than it takes to raise our young. After we raise our young, every successful species since the beginning of evolution, every successful species had a better chance of surviving if it kills off the old. Because in order to shuffle the genes, you have to allow the offspring to breed. And if the old that are more mature and more experienced don't get feeble and unable to perform, they will never allow the offspring to breed. So every successful species, almost every, there are some exceptions and I'm going to mention some, almost every successful species has evolved a way to eliminate the old. And almost every evolutionary biologist will tell you, and my PhD is actually partially in evolutionary biology. So we shut off the telomerase gene. After we're out of our reproductive cells, we shut off the telomerase gene as a pro-death mechanism. It's humans' way of killing off the old. This is, now, so in our ancestors, very pre-human, when we had a rapidly changing environment, it was very important to shuffle the genes as much as possible. Dogs, cats, horses, sheep, pig, and deer, and I probably went that too fast for you guys. They're shaking their heads, yes. Dogs, cats, horses, sheep, pig, and deer are the only animals that we've ever discovered that age by telomere shortening. Rodents, such as mice, they don't age by telomere shortening at all. The studies that Dr. Ron DePinto did, he did them on engineered mice that were made to age like humans. So other animals developed other mechanisms of aging. Mice, roundworms, fruit flies, and yeast, they don't age by telomere shortening. They age by oxidative stress and mitochondria dysfunction. I know this is, am I going too fast for you guys? Okay. And so it's not just humans that have done this. Every species almost has figured out a way to get rid of the old, and they've done it by even different mechanisms. Humans just do it by telomere shortening. That ended up making that whole idea disappear a long time ago too. But now this new thing that Dr. Raphael talked about, genome-wide association studies. has just popped up. And, okay, I'm going to call this the new toy in town. This is, a lot of times new things come along, like a technique called PCR came along one day. Then every lab in the world starts doing everything they can with PCR to publish as many papers as they can as possible. And this is now happening with genome-wide association studies. It's a new toy. Scientists are publishing. You'll find all the studies have like 50 authors because get a lot of people involved, and they're making all these discoveries and publishing them. And you know, 80% of them are going to be refuted later on. It's just too fast, and it's the people doing these studies are not experts in the area that they're studying. They're not experts in, for instance, cancer or heart disease, where they're doing these studies, or other diseases. They're experts in being able to look at genes. So here's one paper, as I said, 50 authors. You can see all these authors down here. I can't even get them all on the screen. This paper came out recently. I think Dr. Raphael showed the same slide. They say long telomeres cause cancer. Another study also came out, and Elizabeth Blackburn refers to these studies when she says telomerase causes cancer. Well, what is genome-wide association studies? And to explain that, I need to first start off with explaining what single nucleotide polymorphisms are, or abbreviated SNPs, SNPs. So when you look at people's chromosomes, now that we can sequence people's whole chromosomes, we see that we take two people, we see that their chromosomes are pretty much 99.9% identical. But every once in a while, we'll find two bases, or one place, where the bases, they're different bases. These G's, C's, A's, and T's are bases in DNA. Such a thing, when they find something like that, that's called a SNP. And so what's happened is that genome-wide association study has now taken this database that's been created of SNPs and started asking questions. How is it associated with certain diseases? So they're just taking, they're screening hundreds of thousands of different people, looking at for these SNPs, and then comparing them to all of their medical records to find out what kind of diseases they had and things like that, to see if there's any correlations between the presence of SNPs and certain diseases. Well, during this process, they found 8 or 9 SNPs that seem to be correlated with long telomeres. People with those SNPs end up having longer telomeres. No explanation as to why, but these little changes in the DNA were somehow resulting in longer telomeres. And then what happened just recently, and which has caused a lot of stir, is that some few papers came out and said that these same SNPs that are resulting in longer telomeres are also increasing the risk of cancer. Okay, so that kind of looks at face value as bad news for telomerase. But I did my own meta-analysis, just for the purpose of this presentation today, because as I said, Elizabeth Blackburn made me mad. I know she's wrong, and I just wanted to go through and prove it in the data. So I did my own meta-analysis. I looked at all studies between 2015 and 2017 that have looked at telomere length and risk of cancer. And I found 13 studies, not just 2. I found 13 studies that looked at SNPs versus risk of cancer. And so when the cancer risk that they're looking at is what they think is long telomeres, 7 of those 13 studies said that long telomeres were associated with increased risk of cancer. Short telomeres, only 1 study said that short telomeres was associated with increased risk of cancer. And then 5 papers said there was no association. Well, that looks like bad news, but I've been in this business for 25 years or more, and I'm a cancer researcher. I know a lot about cancer, and it just didn't make sense to me. Long telomeres cause cancer is just nonsense. But all these studies, they missed a major point, and that is they never measured the telomeres of these people. In all these studies, they didn't look at the telomere lengths. They only looked at the presence of these SNPs. So I thought, okay, let me look to see if there's any studies that have looked at the length of the telomeres, not just the SNPs. And so I found 56 studies in 2015 to 2017 that have looked at, they actually measured the telomere lengths in all these patients and looked at their risk of cancer. All right. 4 of those studies showed that long telomeres cause cancer. But 42 of those studies showed that short telomeres cause cancer. And remember, this is a whole new science. And this is, in fact, if there's ever any discrepancies between what Dr. Raphael and I say, it's only because this is such a new science that people haven't really figured it out. There's a lot of papers that are going to contradict everything you ever find. So there's always going to be some exceptions to the rule. But 42 out of 56 studies showed that short telomeres were what was causing cancer, not long telomeres. 9 showed no association. And actually 2 if you go before 2015, have said that both long telomeres and short telomeres cause cancer. And I can believe that. I mean, everything causes cancer. But they said the only thing that doesn't cause cancer is the average telomere length. So if that's true or not true, I'm not going to dwell on that because only 1 or 2 publications have come out saying that so far. But 42 publications, 42 studies out of 56 have said that short telomeres cause cancer, and only 4 out of 56 said that long telomeres cause cancer. So let me explain what's going wrong here. Why are these people that are doing the genome-wide association studies coming up with the wrong answer? And here's the logic. This is a logic problem they're making. You have a bunch of SNPs. There's only been 8 or 9 that have been identified that are associated with long telomeres. They're associated with long telomeres. Those same SNPs are also associated with increased risk of cancer. But from anybody that's skilled in logic, that doesn't mean that long telomeres are causing the cancer. It means the SNPs are causing the cancer for some unknown reason, and the SNPs are also causing the long telomeres for some unknown reason, but that by no way means that long telomeres are causing cancer. And that's verified by looking at the studies that showed that when they actually measured the telomeres, long telomeres did not cause cancer. It was the short telomeres that did. I'd like to tell you what's really going on with telomeres and telomerase in cancer. This is what a lot of scientists, research scientists, have known for quite some time. Well, first, we all know that every cancer that's ever been discovered was caused by a mutation. Mutations are what cause cancer. Well, so does telomerase, well, I forgot about some slides, important slides here. There is actually no clinical data. There's not a single scientific publication where anybody has, at least not since 2005, and the ones before 2005 have been since retracted, but nobody has done an experiment where they lengthened telomeres in animals or humans and showed that they got increased risk of cancer. In fact, quite the opposite. Every effort to do so has failed, including Dr. Rhonda Pinnell's experiment. Dr. Rhonda Pinnell wasn't trying to show that lengthening telomeres causes aging, or prevents aging. He was trying to show that they would get cancer, and they didn't. The exact opposite happened. I think that's why he left and went to MD Anderson. It's because he was a cancer researcher, but he proved to himself that lengthening telomeres did not increase the risk of cancer. It failed. For every study that's published in the literature that suggests, without data, that telomerase might cause cancer, or that long telomeres might cause cancer, there's at least 10 studies that show that lack of telomerase causes cancer. It's really, we've all had this problem. We have cancer because we don't have telomerase. We get cancer because our telomeres get really short and cause some problems. And that's the bad things are short telomeres. And this is where I was coming back to before. We know that cancers are caused by mutations. All cancers are caused by mutations. And now many papers have shown that short telomeres are a major cause of mutations in chromosome rearrangements, which are just larger mutations that you can actually see in the microscope. But short telomeres actually cause high incidence of mutations. They also cause something called chromotherapis. It's a brand new word for me. I've only learned it in the last 2 years. But this is where people now with sequencing genomes are finding that in cells that have really critically short telomeres, and especially when they have cancer, they have mutations, clusters of like 100 mutations, chromosomal rearrangements, all in the same place. So the telomeres, the short telomeres, are causing the chromosomes to just fall apart, just like the shoelace, the caps on your shoelaces do when they get really short. Your shoelaces fall apart. These mutations are causing the cancers. It's not the only way to get cancer. You can also get cancer from ionizing radiation, et cetera, and things like that, but telomeres are a major cause of the mutations. Did I skip something there? Yeah. Also, telomeres, short telomeres, weaken the immune system. Our immune system is our best defense against cancer, and short telomeres mess those up. Our immune system can't help fight our cancer anymore when the telomeres get really short. So we got to keep telomeres long to keep our immune system strong. Short telomeres cause metastasis. Cancers are caused by mutations. Well, they don't necessarily spread to other parts of the body until additional mutations occur. And those additional mutations are occurring because the telomeres are short. I mean, if we could lengthen telomeres in cancers, that would probably give our cancers a less likelihood chance of metastasizing. And we don't know that yet because nobody's done an experiment like that yet. It's all, as I said, we're still in research. And then short telomeres allow cancers to survive whatever you hit them with. We've all heard about cancers, chemotherapy, getting rid of a cancer and the cancer coming back. Well, they're coming back because of additional mutations that made them survive the treatment that you were hitting them with. The short telomeres are increasing the mutation rate so high that the cancers are coming back because of short telomeres. I believe we need to keep the telomeres long and do something to keep them long so that we can improve our ability to fight our cancers. Now, I already talked about my own meta-analysis, but there's been lots of meta-analysis now done where there's so many studies, and I only looked at from 2015 to 17. Now this is a study, I think the year is somewhere on there, but I don't see it, but it was before 2015, where they did a study, looked at all the papers that were looking at telomere length in cancer, and came up with the conclusion that short telomeres are associated with increased risk of cancer. This is old news. Elizabeth Blackburn should know this. Now another study just came out just 3 weeks ago, saying the exact same thing. When people actually measure the telomeres and just don't play with this new toy called genome-wide associated studies, you actually see that short telomeres cause cancer. And I'd like to go in one step further to make this kind of more obvious that long telomeres and telomeres don't cause cancer, by talking about the fact that there are some animals that don't have any detectable aging. This is something new, but the first one that was discovered was lobsters. Lobsters. I mean, nobody cared how long an animal lived until the time of Darwin. Then people started saying, well, let's see how long they live. But the only way to see is to be there when they're born, put them in a cage or aquarium, and then watch to see how long they live. And 150 years later, we're still seeing some of these animals are still alive and healthy and kicking happily ever after. Well, a study came out showing the reason why lobsters live so long is because they have telomerase produced in every cell of their body, not just their reproductive cells like humans do. They have it in every cell of their body. Well, Elizabeth Blackburn would tell you these lobsters must get cancer pretty bad with telomerase in their cells and having long telomeres, but in fact, they don't. They rarely get cancer, and they rarely get other diseases. And it's not just lobsters. This has now been shown with clams, tortoises, humpback whales, fish, and some birds, or some fish and some birds. All these animals have been shown to have telomerase produced in all their cells. They don't have any telomere shortening. They have no detectable aging, and they rarely get cancer and other diseases. Charles Darwin had a pet tortoise named Harriet. She had just died recently at 180 years old. She didn't die of aging, she died of some virus infection. Humpback whale was discovered with a harpoon in it, and they carbon dated that harpoon, and it was 130 years old. These animals are living a long time. And we don't know how long they live because most animals don't have like rings on a tree that we can count to measure how old they are. The exception is clams. Clams have these stripes on their shells. They get a new stripe every year. Well, lo and behold, people are now finding clams over 500 years old, all because they have telomerase produced in all their cells and they have long telomeres. They're not getting cancer. They're not getting other diseases. They're living long, healthy lives. So, bottom lines, telomerase does not cause cancer. The lack of telomerase causes cancer. We've known beyond a shadow of a doubt for a long time that this is true. It's something that we have to solve. And for 25 years, this has been one of my missions. Because when I was a cancer researcher and I moved into telomere biology, I thought this is a way to cure, prevent, fight, whatever you want to call it, cancer by making telomeres long. And that's why I get mad when somebody like Elizabeth Blackburn tries to say that's not true. The opposite is true. I've just spent 25 years trying to find a cure for cancer by lengthening telomeres, by inducing telomerase. We need to keep telomeres long. That's the bottom line. If anybody would like to see any of these papers that I talked about, including all of those 56 studies or 13 studies on the genome-wide association studies, you can get these papers full text at my company website, which is www.sierrasci.com/papers. I just want to end the nonsense. I want to make all the data available to everybody that really is still questioning this fact that telomerase might cause cancer. And